kind introduction. And first of all, I would like to thank the organizers, like Julien and many people who are helping. Multidisciplinary meeting. So today I'm going to, we are in a Bioinspired lab in Marseille, belonging to the Institute of Movement Science. And this institute belongs both to CNRS and the University of Aix-Marseille. So the institute is located near the Calanque in Marseille, for those who know the nice place here. My first remark would be that 100 years ago, Louis Blériot flew over the English Channel, as you may know, from Calais to Dover, not very far away to the north of Nantes. This was in July 27, 1909. This was a big challenge in those days, 100 years ago. 100 years ago. But 100 million years ago, there were already plenty of ace flyers on Earth. And these were the insects. I remember what Sten Grillner said this morning. You mammals were hardly born in these days. So this talk will deal with visual-motor integration, basically. And we attempted with my colleagues to close the loop between motion, visual sensors, and environment over the last years. And first of all, I would like to show you what optic flow is and what optic flow sensors are, and ask whether we can rebuild them in a way, perhaps in electronics or in some other man-made technology. And then I will deal with how can optic flow sensor help avoid the ground, and how could optic flow sensor help avoid lateral obstacles. So the bioinspiration in that case is twofold. As far as the optic flow sensors are concerned, the inspiration comes from single neuron recordings from the eye of flies. And as far as optic flow-based autopilots are concerned, the inspiration comes rather from the behavior of flies and honeybees too. So I want to stress what Jérôme already said, in fact, that if we are taking inspiration in biological systems to build bio-inspired sensors and bio-inspired autopilots, we are in fact, at least we are in our laboratory, mainly interested in the reversed arrow, namely in how far these elementary robots that we are building can help us to raise new biological hypotheses, raise new questions. And of course, this is a very important point because, in fact, we are constantly applying from biorobotics to biology, and we try by iteration, in a way, we attempt to improve our knowledge of how the animals work. So, of course, although I am going to deal with vision and with the compound eye that these insects possess, I don't want to miss the point that these fancy animals are not only equipped with eyes, but they have also many, many other sensory modalities, particularly the antenna with their olfactory and wind receptors, and many sensors, many mechanosensors on the head and between the head and thorax to measure, for example, the relative position of the head with respect to the thorax, an important parameter. But also on top of the head, you always have three tiny eyes, which are called the ocelli. And these ocelli, which are shown in the magnification here, magnified view is scanning EM here, have been shown, at least in dragonflies, but 20 years ago, to be kind of horizon detectors. In other words, the frontal ocelli, which is looking forward, will adjust more or less the pitch of the animal with respect to the horizon, whereas the two lateral ocelli will adjust the rolling of the animal with respect to the horizons. Among the other important sensors that we have in this system are the two halteres, which are inertial sensors, which you see here. One is here and one is here. And these tiny masses actually vibrate the same frequency as the wing, but in antiphase with the wings. And these halteres have been shown 60 years ago to act as miniature gyroscopes. They are able to sense the Coriolis force that is produced when the animal is yawing or pitching or rolling. For this, you have plenty of mechanosensory sensors here at the base of the whole ears, which will which will report the brain about these Coriolis forces and will attempt to stabilize the animal by mechanoreceptive feedback loops. Now, of course, I'm going to deal mainly with the compound eye, with this beautiful structure, with many facets. In fact, each facet, you see a reflex on each facet here. There are, let's say, approximately 3,000 facets in each eye, and each one is a tiny lens of diameter 25 microns and focal length 50 microns. And you see that basically you have 3,000 pixels in each eye. Remember that in our own eye, we have 126 millions of pixels in each eye. Here you have only 3,000 pixels in this eye and 3,000 pixels in that eye. Very low resolution, and moreover, these pixels are organized basically in the 4 pi steradian, even more than that, because there is some binocular overlap here in the frontal part of the head. Now, the fly characteristics, our capabilities are just amazing, because with a mass of less than 1 gram and a size of less than 2 centimeters, a maximum speed of 10 meters per second, it reaches yaw rotational speed of up to 3,000 degrees per second, and yet the signal processing is done by only 1 million neurons at most, among which you have 50,000 photoreceptor cells in the eye and approximately 50,000 olfactory cells located in the antenna. And all of them converge in the brain in order to drive the descending neurons that go through the neck of the animal and will drive indirectly the indirect flight muscles for the legs, and the leg muscles as well. So these animals are capable of obstacle avoidance, as we will see. They are capable of hovering. They are capable of accurate landing, and they are capable of smooth pursuit. Now, you may wonder what they are pursuing, what they are tracking. So in the next film, a very short film, which was produced last year by Weingart, I want to impress you by showing you how the male Diptera is following the female at high speed and doing a lot of side slip. You will see a female that by chance sat on a flower, and on that day there was a lot of wind, so the flower is going from right to left at a high speed, and you will see what the male fly is doing. You see the female here and the male, which constantly pursue by doing side slips at a very regular and high speed. So see, this is one of the prowesses of the fly. And so we are going to see first how insects may navigate in this 3D environment. And first of all, and this is a kind of recalling former works that we did at the laboratory, mainly in the 80s, analyzing the optic flow sensors and how we, in the mid-80s, how we could switch or emulate these optic flow sensors into tiny electronic optic flow sensors. And this was, by the way, the start of our biorobotic activities in the mid-80s. So everybody knows what the translational optic flow is. Everybody has been at the window of a train, and if you look perpendicular to the train direction, you see something like that. But basically, what you have to consider is that the optic flow is the angular speed expressed in radian per second or in degree per second, at which any contrasting feature in the environment will move past the eye. And this tiny video, which was taken by Julien between Marseille and Cassis in his car, just shows you what you can see. And what you will notice is that those things which are far away, like the mountains, these contrasting mountains in the far, they move at a very low angular speed, whereas the rocks, which are here close by, they move at a very high angular speed. So there's a way here to measure something, and at least to measure the optic flow. But in fact, in order to measure the optic flow, which is represented here by this vector field, representing what you would experience if you would be heading towards the point O. Suppose you are walking or flying towards the point O. Then those flowers or those grasses, which are on each side of you at 90 degrees, they whiz by at a very high speed, high angular speed. Whereas those things which are close to your heading direction and far away in the front, they give rise to very small vector of the optic flow. So this is the difference between those objects which are far away and which are far away at a great distance, and those objects also which are near the heading direction, where the phi angle is the direction of the object with respect to your heading direction. And you see that if phi is equal to 0, that is, if there is something here in the neighborhood of your heading direction, this will give rise to hardly any optic flow. The difficulty, this is called the pole of the optic flow field, and it is very difficult to measure the relative motion between yourself and these objects in the environment when they are located near the pole of the optic flow field. But one thing is sure, is that if you want to measure the optic flow, it requires motion-sensitive neurons. And we know that we humans have these motion-sensitive neurons, not only in the visual cortex in V1, but also in deeper structures, especially in the so-called mediotemporal visual area, in MT, which is a rather small and deep structure in the cortex, rather difficult to analyze. Now, these motion-detecting neurons have also been found in flies, and they are known to a great extent in flies, in fact. You see a cross-section through the head of the fly here with the facet lens, which you showed before, and to which you saw a reflex, and this gives rise to an ommatidium. An ommatidium is a small eye with a few receptor cells located in the focal plane of this tiny facet lens. And these photoreceptor neurons pass their information to second-order neurons, and then to third-order neurons, and to fourth-order neurons, down to the level of the lobular plate, where we find this magnificent set of motion-detecting neurons. They are approximately 60. They number 60 neurons, which are large field neurons, the dendritic field of which is perpendicular to this plane. So we can access, actually, these motion-detecting neurons by making a tiny hole here in the back part of the head capsule with a split of a razor blade, and we can impale with a microelectrode. And in such an experiment, an ideal experiment, when you would come from the back of the head, you would first encounter vertically sensitive neurons. From these 60 neurons, I'm going to show you 23 of them here. So the microelectrode, they are shown in an exploded view. The microelectrode would first encounter vertically sensitive cells like V1. One of these VS cells, V2 cells, the H4 cells, then horizontally sensitive neurons. You see the direction of the arrow represents the optimal direction where you have to move something in order to elicit a response in this neuron, and then move again horizontally sensitive neurons, the three famous horizontally sensitive, the north, the equatorial, and the south HS cell, and the centrifugal. So this is only part of these 60 neurons. And in the mid-80s, with my colleagues Alexa Rieler, we chose to record from that neuron. I should insist on the fact that these neurons can be found again and again in every fly. Whether you open the head in Tokyo or here, you will find these neurons exactly the same at the same place. And we can record from them, and we know in real time that we are recording from H2 or from H1 due to their particular characteristic of the response. So if you record, for example, from this H1 cell, you see something, you see that it responds like a machine. If you move something in the visual field on the screen of an oscilloscope or a computer, and if you move this pattern in the direction from the back to the front on the head, on the eye, you see that you get a beautiful increase in the spike frequency. These neurons are spiking like many vertebrate and invertebrate neurons, and they give you here 130 hertz, 130 impulses per second. And as soon as you stop the motion, the neuron goes down to the resting activity, if it may be 15 Hertz. And if you move the pattern in the wrong direction, you see that the reaction, the response is even inhibited. So this is really a machine-like response. You can record from these neurons during days and nights, if you like. The problem with these neurons, and in order to understand the underlying response, is that H1, like the other 59 neurons, actually collects input from thousands of tiny neurons, which we call the EMDs, the elementary motion detectors. And in those days, we attempted first to analyze the operation of these EMDs. This is much more difficult because you have to go down to a very fine stimulation. And the idea was actually to stimulate single photoreceptor cells despite their very tiny diameter of 1 micron. So in those days, we had discovered, in fact, that the photoreceptor cells are fluorescent. So in response to a very strong blue light sent in this direction through the facet lenses, you can see the photoreceptor cells, and all of them, all the peripheral cells, 1, 2, 3, 4, 5, 6, emit red light, whereas the central one emits sometimes green light, sometimes nothing, and sometimes red light, another red light. These two cells, they're very fascinating. We know now that they are done for color vision. And the central cell, number 7, here in the center, is actually decomposed into 2 tandem cells, which means that light must first penetrate into this light guide, the cell number 7, in order to reach the cell number 8. Whereas the 6 other cells are done in particular for motion detection. So if we want to analyze the process of motion detection, we have to deal with the red cells here, containing a special visual pigment and very sensitive cells which have an ultraviolet sensitizing pigment, as we showed in those days. Now, if you poke a microelectrode within any of these cells, you get, in fact, a beautiful electrical response. Normally, the cell's potential is minus 70 millivolt, and as soon as you shine light during these 500 milliseconds, for the very high light in this particular case, you can get up to 60 millivolt depolarization. So each one of these thousands of cells give basically the same depolarizing response, which is called the receptor potential. Now, the idea of analyzing motion detection at the elementary level was this. We know that these 7 cells here in the focal plane of the facet lens, each of them look in a different direction in space. And think of what motion is. If you move something in front of my eye, this contrasting border here moved in front of my eye will successively excite many rods and cones. So there's a sequential excitation of my photoreceptor cells. And what the motion detecting neurons do is to analyze the sequential stimulation. So what we aim to do is, in fact, to stimulate first this cell, and then 50 milliseconds later, we would stimulate this cell, so to speak, to simulate a micromotion that would take place in the tiny visual field of that ommatidium. So to do that, we had to switch to a special instrument that we call the microscope telescope, because it is, in fact, a hybrid between a microscope and a telescope. There's a switch here somewhere that you can push, and you can transform this microscope into a telescope. With the microscopic part, you can access the surface of the eye, and you can select a given facet, 25 micron diameter, focal length 50 microns. And you know that behind this facet lens, you have the 7 photoreceptor cells. And by means of the telescopic part of the instrument, we can actually select, we see these 7 cells, and we can select 2 of them, and we will now stimulate them one after the other in order to simulate a micromotion. And if you do that, if you record from the H1 cell and the sequential stimulation of these 2 photoreceptor cells, you see something, in fact, quite beautiful. You see the response of this sequence gives you a burst of spike. You do the experiment again. Whenever you do this sequential stimulation, you get similar bursts of spike. Now, if you reverse the phase, first starting with the cell number 6 and then ending with the cell number 1, 50 milliseconds later, as shown here, you get hardly any spike. You see that we are here close to the mechanism of motion detection. And it took us approximately 2 years to come to a model of this system. And I don't want to spend much time on that. Just would like to show you one example of this response. Now here, the spikes are plotted in terms of the instantaneous frequency. So you see that if you do again this experiment, first starting with the cell number 1 and then ending with the cell number 6, as shown here, where these 10 millisecond spots are far apart from each other, you get a beautiful increase in the spike rate, up to 80 hertz, perhaps. Now, if you reverse the phase, you see that the resting frequency is nearly inhibited at that place. So there seems to be more or less a symmetry in the response. Now, needless to say, very, very soon, in 85, 86, we had our first electronic motion detectors inspired by these electrophysiological data on single neuron recording and the single photoreceptor stimulation. And what we did, in fact, is that basically we had 2 photodiodes, and imagine that these 2 photodiodes look at different directions, separated by an angle delta phi. In flies, the angle between 2 facets, or in flies, in insects in general, is always in the order of 1 degree, 1 degree, 2 degree. In the fruit fly, Drosophila, that we show this morning, it is 5 degrees. Delta phi is 5 degrees in Drosophila, 1.5 degrees in housefly, and so on and so on. And now suppose something moves, a contrast will move in this direction. Obviously, it first hits this channel first, and a certain time later, maybe 50 seconds later, it will hit the second photodiode. Now, what is done here is something, in fact, quite biomimetic. It reproduces basically the processing of the first synapse in the fly retina, between the photoreceptor cells and the second-order neurons. And then, on both channels, we have simply a threshold circuit, something which is quite feasible in a nervous system. Then we have here one single pulse generation on each channel again. But then here, this first pulse in channel number 1 is used to generate a long-lived exponential decay. And this exponential decay is simply sampled by the second pulse arriving in the channel number 2. So you understand that by the time the contrasting border arrives here, let's say 50 milliseconds later, for example, you will sample the exponential at a height which depends on delta t. So if something moves very fast, so delta t is small. If delta t is small, you will sample the exponential at a high value. In other words, what you get here at the output is something like the optic flow, omega measured optic flow, which is more or less equal, or more or less proportional to delta phi over delta t. So that is how we built these motion detectors early in the 80s. And over the years now, we have built several versions of these elementary motion detectors. This was one we had in '89. And at the same scale, those we built more recently based on low temperature coal-fired ceramics in collaboration with Oulu University in Finland, and here in collaboration with the Center for Particle Physics in Marseille. We had this integrated array of adaptive photoreceptors followed by the elementary motion detecting circuits based on a micro FPGA, reprogrammable digital circuits. That's how we started, in fact, in this field of biorobotics, because we felt we had something valuable in our hands, namely elementary motion detectors, and we wanted to go further and to try to see how an animal really, whether and how an animal could use them in order to navigate in space. So this is the curved compound eye that we built early by the end of the 80s. And you see here that basically between any pair of facets, 2 facets again are looking in different directions in space, are sampling the visual space in different direction, and between any pair of 2 facets, you have one such elementary motion detector. And it is based on this curved compound eye that we built the RoboFly about 20 years ago. Basically, you remember this formula of the translational optic flow. Now imagine that this robot with this compound eye here, or a human, or a fly, would be moving here at a speed v0 in this direction. If at the azimuth phi, the eye encounters a contrasting object, and if on board this eye you have a means to measure the optic flow omega, at which this point P seems to move in this direction when you move in this direction, then the formula just simply tells you that by knowing omega and knowing your speed, you can retrieve the distance to the obstacles. And you can do that in a panoramic way, and you can do that for all obstacles present in the world. And so this is based on this feature that this first RoboFly or RoboMouche operated. See, the compound eye is mounted here, and this is just a target tracker. It actually tracks a lamp in the corner of the laboratory where it has to go. This is the heading direction of the robot, but primarily the robot has to avoid the obstacles it will encounter on its way to the target. Now, perhaps we can summarize the operation by saying by moving forwards, the RoboFly generates its own translational optic flow to which its compound eye will be sensitive by means of the array of elementary motion detectors. And the second point is that from the measured optic flow, the robot can determine the distance to obstacles and therefore avoid them and drive around them autonomously at high speed. So the robot runs at relatively high speed in a completely autonomous way. We may see perhaps some of the behavior if it works. So you see that basically this corresponds to one horizontal section through the compound eye of a fly. And the robot, see that it's going to its target, but while going to the target, it primarily has to avoid these randomly placed obstacles. And it does that exclusively by means of these optic flow sensors. Now good, this is a good point, but in spite of the early success of this optic flow-based robot, we were not especially happy. And the reason for this is that this robot in a way is cheating. Why is it cheating? It's cheating because obviously there's an optical encoder on the wheel which delivers the speed v0. And of course, if the robot can measure omega and it knows it's v0, it can retrieve the distance to the obstacle. But what about insects that fly? Flying creatures in general, terrestrial animal can estimate its ground speed, and so does the RoboFly. But what about aerial insects and birds, which lack any contact with the ground? This is a much more difficult stuff, and we have been dealing with this over the last 20 years. With various robots, in fact. I just want to remark that to prevent crashing, conventional aircraft and spacecraft and autopilots need to measure the ground speed, which can be done by Doppler radar or GPS, need to measure the ground height based on a radar or laser rangefinder, and they usually need to measure the descent speed with a variometer. And the question is, do flying insects really need such sensors? And if not, how do they avoid crashing on the ground all the time? That's the big problem. And this brings me to the second part of my talk, where we tried to have a biomimetic control scheme for ground avoidance. Now, imagine an insect or a flying creature, a bird, flying over this field. Very complex, so they see practically nothing on the right and the left. They see mainly the ventral optic flow. And you can have an idea of what they see. Try to imagine the processing, how fast the processing must be done in order to analyze the optic flow coming from the ventral ommatidia of the insects. it's not a small problem. But try to make it simple. If you have a fruit fly like this one, as seen from the side, flying over a random terrain like this one, well, something you can state is that the ventrally located ommatidia, if the animal is flying at the ground height h and at the ground speed vx, I say ground speed and not air speed, then the optic flow omega in radian per second is equal simply to vx over h. This is rather simple. And we had for this control scheme several sources of inspiration, and the first one comes from Kennedy's optomotor hypothesis, which dates back to 60 years ago. And what Kennedy stated in those days was that flying insects, to his mind, have a preferred retinal velocity with respect to the ground. We would say now they have a preferred optic flow with respect to the ground, because, in fact, the optic flow is transferred in a linear velocity on your retina by means of the optics of the eye, whether you have a compound eye or a camera eye like we humans. So this is an important point, maybe a hypothesis which would be worthwhile. Now, by the end of the 90s and in year 2000, Srinivasan and his colleagues in Canberra showed that even when a bee is landing, you see that the horizontal, the height of the animal is going from 25 centimeters above ground down to zero. And he noticed by filming the trajectory of the fly that when the height decreases, the speed decreases as well. So that when so doing, when the bee is landing on a flat terrain, you can say that Vx over h remains constant, much in the same way as this hypothesis of Kennedy 60 years ago. Now, this does not tell us much because, of course, to make your optic flow constant, you can fly, for example, at a height of 1 meter height, at 1 meter per second, and you will make sure you have a 1 radian per second optic flow. But you would get the same optic flow if you fly at 2 meter high and at 2 meters per second. So this doesn't actually tell us how the bee managed to land in this way. There are still basic questions that remain open. And one of these questions is, which flight variables do insects actually measure? Which flight variables do insects control? And what is, after all, the logics behind the flight visuomotor control system? That's what we attempted to deal with in recent years. Now, you have seen this left picture already, but what the right-hand side shows you is that if you have a lens like the tiny lens of an ommatidium, for example, and 2 photoreceptors behind, and if you have an elementary motion detector behind, then you can access a measured optic flow. And so you can access this optic flow value, which means that you can access the ratio between Vx and h. Of course, this will not tell you your speed. It won't tell you your height either, but at least it tells you the ratio between these both flight variables. And this is how we designed this octave autopilots with Franck Ruffier and Julien about a few years ago. This is called Octave, Optic Flow-Based Control System for Aerial Vehicles. And basically, Octave attempts to regulate the optic flow measured, omega measured. That is, to maintain the optic flow constant and equal to a set point. This is what the word regulate means, in fact. And Octave does so by controlling the lift. Octave is a feedback control system that we call the optic flow regulator, and the sensory feedback is simply provided by an optic flow sensor like the one we analyzed in the fly. Octave, so I want to describe how this control system works. So the main point is that there is no altitude servoing, and there is no ground speed servoing either, for one good reason. The animal is probably not able to measure its altitude and probably not able to measure its ground speed. So you could hardly build a servo system on this basis. Now, let me show you on this right-hand side, again, a fruit fly. And the fruit fly has a given body orientation, and it has a given mid-flight force orientation, which can be quite different, in fact, from the body orientation. And this mid-flight force vector can easily be decomposed into the forward thrust along the surge axis and in a vertical lift along the heave axis, which will allow the animal to stay aloft and to rise if needed. Now, if you pitch the animal forward from its, let's say, hovering position, a few degrees forward, this is what is done here, you pitch forward, then you get some forward thrust. And via the surge dynamics, you immediately will get some airspeed, and from the airspeed, you get some ground speed. Now, remember that the ventral optic flow is the ratio between these two values. Now, the vertical lift via the heave dynamics gives rise to the ground height, and it will operate this ground height. Now, the ratio here in this nonlinear box is omega, which now can be measured by means of these optic flow sensors, like the EMD of the fly, and the output of the optic flow sensors is constantly compared to the optic flow set point. If there is some deviation and some error signal, epsilon, via the controller, it will move the h, it will move the height of fly so as to retrieve the original optic flow and make it constantly equal to the set point. Now, this system is in fact quite interesting and lead to insect-like behavior. We have helped ourselves to analyze this system. We have implemented it on a tiny helicopter, 100-gram seeing helicopter. Basically, there are two things at least to be said about this system. It has a rotor, and the lift depends on the rotor speed, simply. There is no cyclic pitch or collective pitch in this one. The second thing is that there is a tiny eye, a very elementary eye, let's say one omatidium with two photoreceptors, and behind the two photoreceptors, you have one single EMD. And the eye is always oriented towards the ground, and it is sensitive to the randomly oriented ground and randomly contrasted ground. Now imagine, for example, that you pitch the robot forward by a few degrees, 1, 2, 3 degrees. You immediately get some forward thrust, you immediately get some ground speed, so you have a given Vx. And if you have Vx, it means this is going to you get some imbalance of the optic flow, which will be different from the omega set. You get a positive error signal that will drive the H immediately, giving rise to a kind of automatic takeoff. As soon as you pitch forward the insect or the robot must rise also into the air as a consequence of the feedback system. This is what is shown here as a recording from this tiny helicopter. You see that between arrows 1 and 2, the helicopter was slowly and gradually pitched forward up to these few degrees pitched forward. So there was a gain in the ground speed, and because there is a gain in ground speed, there must be a gain in height. In other words, there is an automatic takeoff. Now at some time, due to the drag, the ground speed remains more or less constant, and for that same reason, due to the feedback, the height remains constant as well. Notice that during that time, and in spite of the fact that there is a dramatic increase in ground speed in meter per second here during this phase, the optic flow here is measured is nearly constant. And the actual optic flow, which can be calculated at each abscissa by dividing the ground speed by the ground height, is represented here. You see that also it is basically constant. This is the purpose of this complete feedback loop, to make the optic flow constant. And this is how it gives rise to the behavior of the animal. Now, during these two arrows, we did exactly the contrary. That is, slowly and gradually, the body was raised to the vertical. And naturally, you get a decrease in speed. And because you have a decrease in speed here, you get a corresponding decrease in the ground height. Now, Franck Ruffier, in the vision session tomorrow afternoon, will explain you in more detail why, in fact, in the last final approach, the landing phase, the robot lands, in fact, at a constant descent angle. And this is all the more interesting since in the recording from Srinivasan about honeybees, they showed that when bees land on a flat terrain, they land at a constant descent angle. So in a way, this system can explain or may explain the constant slope landing of honeybees. Take another example, for example, terrain following. Suppose that the animal or the robot would encounter a slowly climbing terrain, a rope. Then it means that h, the local height, is gradually reduced between the robot and the ground. If this is reduced, it means that omega is too high. It's too high with respect to omega set. So again, the deviation here, the error signal is positive, and it will raise the height of fly so that finally there will be terrain avoidance. And this is illustrated in this video. You see that the helicopter is rising over this ramp, or should be rising over this ramp. Sorry, perhaps. Let's try again. It's rising over the ramp, and immediately up after the ramp, it comes down and can do that during hours if there is enough energy. Sorry, you can see the results of these experiments as recorded. If we launch the robot at a low speed, or at the speed of 1 meter per second in that particular case, then you see that the robot is rather really hugging the terrain, the slanting terrain. If we launch the robot at 2 meters per second, a high speed, it will fly at a higher height. So you immediately see that by means of this optic flow sensor, the altitude varies automatically, and you get automatic ground avoidance. And the higher the speed, the higher the flight. And the altitude attained is safe, you can say safe, because it increases suitably with the ground speed. Now, in fact, this is quite interesting because this octave autopilot may account for the terrain following abilities of dung beetles, as shown by Steiner in 53, also of migrating butterflies in canyons, as shown by Williams in 65, other migrating butterflies in dense forests, shown more recently by Srigley and Oliveira. So it seems that this system actually captures much of what is on board the insects. Just to show you how reproducible all these trajectories or flight paths are, 5 trajectories are shown in each time, and you see that they are, whether you are dealing with the automatic takeoff, automatic terrain following, or automatic landing, you always find more or less the same thing. And Franck Ruffier can tell you that we actually never had any crash in the laboratory. Although we are using only a single optic flow, and we know that flies do not use a single optic flow sensors, but they use thousands, even for the ventrally oriented optic flow sensors. Now, an interesting point which I have not discussed thus far is that this system, without any change, also leads to a sensible reaction to headwind. By headwind, obviously, your ground speed is reduced. There's a brake due to the wind. But if the ground speed is reduced, and since there is this feedback loop that acts upon h, so the robot must descend, because h is constantly made proportional to the current ground speed. And indeed, here in that area, being -4 and 0, the robot faced a slight headwind and you see that its altitude gradually decreased, nearly close to the ground. And in the case of a very strong wind, very strong headwind, the robot even went down, and the strong headwind basically led to a forced landing. But again, this forced landing occurs under sensible conditions because the horizontal speed at this point is 0, and the vertical speed at this point is 0 as well. Now, the advantage of this system is that measuring the optic flow appears to be sufficient, and there is no need for an explicit knowledge of ground speed and no need for GPS or Doppler radar. There's no need for an explicit knowledge of ground height, and therefore there's no need for a radar altimeter, and no need for an explicit knowledge of descent speed, no need for a variometer. In other words, the optic flow-based autopilots saves major avionics sensors, which are reputedly heavy, costly, and power-hungry. But at least it would need photons and contrast. Contrast and photons, at least in some spectral window. Turns out that insects see between 300 nanometer in the near UV up to 600 or 700 nanometer in the red. But you could imagine exactly the same system with the same optic flow sensor operating in the far infrared up to the terahertz, if needed. And this system is based on simple processing, which is well within the neural repertoire of the insect brain and easy to implement on board synthetic creatures, in particular on board micro-aerial vehicles. Now, the principle might be applicable to micro-air vehicles, micro-space vehicles, and perhaps also to benthic micro-underwater vehicles, which would fly or swim close to the ground of the sea. Now, also, if you look at poster number 1, and if you hear Franck Ruffier's talk tomorrow afternoon in the vision session, you will see how these data can also lead to optic flow-based moon approach. This is a simulation. I don't want to go into the detail, but this is the moon. This is the Pangu simulator of the moon, which was given to us by the European Space Agency within the framework of a contract. And you see that basically there's a given law that is given for the pitching of the moon lander. So it is raising progressively down to this point. So the moon lander arrives at approximately 2,000 kilometers an hour, and in the end, it ends up with only 5 meters per second at a height of 10 meters above ground to approach the final landing approach, which is always done by laser in the moon landers. Now, the main point here is that the optic flow has remained constant throughout the landing from the very high speed originally down to the point close to the ground. Now, in the same way, I want to show you that you can make a similar biomimetic control scheme for lateral avoidance, as was shown in recent years in our laboratory by Julien. So the inspiration this time came from the famous work of Srinivasan in Australia, and many of you know that when a bee is entering a tunnel like this one, usually the bee centers along the tunnel. And Srinivasan, 20 years ago, did a nice experiment where he, for example, he moved the right wall here. And what he observed is that the bee would come closer to the moving wall. And in that case, he moved the wall in the other direction, and he moved the wall in the other direction, and you see that the honeybee is like frightened and goes away from this and very close to the static wall. Now, to explain these reactions, Srinivasan obviously said that actually the bees normally show a centering behavior, as shown on the top corner in A, and he said that they do not react to the distance proper, but they rather react to the lateral optic flow. Now, he put forward the optic flow balance hypothesis, saying that if the bee, for example, travels in the middle of the corridor here, it is because it simply tries to balance the optic flow it receives on the right-hand side and on the left-hand side. So we have done these experiments again in recent time in larger tunnels, And this is the work made by Julien on the flying honeybee in a similar tunnel, but much larger, a 1 meter broad tunnel. And indeed, if you launch the bee in the center and have the reward in the center here, the bee more or less fly centered along this tunnel, as Srinivasan said. But if you launch the bee on the left-hand side, with the reward on the left-hand side as well, you see that after the training session, the bee comes into the tunnel and they stay here and they do not cross the tunnel by being centered. Now, for the reverse case of the entrance on the right-hand side, they stay here on this right-hand side. So this seems to be at odds with the Srinivasan hypothesis, especially in that case where we removed that wall, the left wall. And you see that it is as if the left wall will not count much for the bees. They stay centered and they are basically fly at the same distance from the wall as they were flying in the presence of that wall. As if here, the absence of presence of the left wall would not count at all. Now, it's nice to analyze these results. And you can say basically that the bee obviously has a left optic flow, which is equal to its speed divided by the distance to the left wall. It has a right optic flow, which is simply the speed divided by the distance to the right wall. And if you analyze these values, you observe that in the first case, when the honeybee flies centered, indeed, it has more or less the same optic flow here, 140 degrees per second. So the radians per second were converted here. Now, when the bee flies on close to one wall, you see that whether it is the left wall or the right wall, it has a quite different optic flow from the previous case, and more or less these two optic flows on the right or on the left are more or less similar. If you remove the wall, the optic flow is a bit higher due to a higher speed of the animal. But one point is important, is that on following a wall, you can say that in these two cases, the bee maintains an ipsilateral optic flow of something like 230 degrees. The conclusion from these behavioral experiments are this. In addition to the centering behavior, bees can exhibit wall-following behavior. And the optical balance hypothesis from Kirchner and Srinivasan 99 cannot account for this wall-following behavior. We instead consider that bees regulate, that is, maintain constant, their lateral optic flow with respect to one wall. In the same way, they were regulating their ventral optic flow from the ground with the octave system. So our working hypothesis is in fact that bees control their clearance from the walls by means of a lateral optic flow regulator that produces side thrust. Now, in recent time, it has been shown very beautifully how side thrust can be produced. You have seen the case of the male hoverfly that was producing side slips close to the female fly. Now, in another case, namely an Hymenopteron, which is the sand bee, Jochen Zeil in Canberra and Norbert Bödecker from Bielefeld recently showed how this side thrust is produced. This is shown in a sound wasp and now at high speed, 500 frames per second. And they showed that side thrust is in fact produced by rolling. What you will observe in this very short video is that basically the sound wasp, which is more or less hovering here over its, the burrow nest, the sound wasp maintains its head basically fixed in space, and it rolls the thorax to the left or to the right, depending on the side slips it wants to make. A beautiful roll and breaking with another roll, while the head remains practically fixed in space. So you see these beautiful side thrusts that are produced by rolling. And it looks as if forward and sideways motion were uncoupled, mechanically uncoupled. And this would explain, in fact, some earlier experiments by Srinivasan and Kirchner, who showed in 20 years ago that when a bee is entering, in fact, the tunnel here, its body, this is the head of the honeybee, and this is the direction of the body. The direction of the body remains more or less parallel to itself. So there seems to be an uncoupling, mechanical uncoupling, between the forward speed and the lateral speed. And so this gave rise in our laboratory to another honeybee, which is much more simple. In fact, we didn't want to bother with rolling a thorax with respect to a fixed head. So we used a very elementary micro-air vehicles, which flies at about 2 meters or 5 meters above the ground. It's a hovercraft. But to this hovercraft, we added 2 eyes, which are laterally oriented to the right wall of the tunnel and to the left wall of the tunnel. And especially, we added 2 side thrusters to the rear thrusters. Now, the 2 rear thrusters push the machine forwards and getting the ground speed, the forward ground speed, while the 2 lateral thrusters produce the lateral speed, Vy. You have an example here showed that we don't need to see that, perhaps. So the lateral optic flow regulation autopilot that we devised is in fact this one. First, you have the 2 rear thrusters which push the device forward, getting the forward speed. And this forward speed is involved both in the left optic flow and in the right optic flow. And the side control signals via the integration will give rise to a given positioning within the width of the corridor. Namely, it gives rise to the distance to the right wall and the distance dl to the left wall. You see that these 2 things are completely intermixed. And well, the output of these 2 systems, as you heard before, is simply the right optic flow and the left optic flow. And what the first control loop is doing is that it takes the maximum of the optic flow, and it is based on this maximum between the right optic flow and the left optic flow. It is this signal that will be now compared to an optic flow set point. We call it the sideways optic flow set point. And the deviation from that point will drive this loop and will position the robot in the width of the corridor. That is, it will determine dr and dl. So this is for the side control, the lateral positioning within the tunnel. And now there is a second loop that will act upon the 2 rear thruster to define the ground speed. And this loop operates also only on these 2 lateral optic flow, but in fact it operates on the sum of these 2 optic flow. And it is the sum of the right and left optic flow that is compared to a second optic flow set point. We call it the forward optic flow set point. Now the deviation here via the forward controller and the forward dynamics will define the current ground speed. And so you have 2 intricate loops which are intermixed here in defining the speed and the distance to the walls. Now, if you do the simulation, as was done here by Julien and Julien, you see that if the robot is entering here in this tapered tunnel close to that wall, it will enter the tunnel and it will gradually break. And here from that point on, after the constriction, it will accelerate as shown by these curves. This is the speed represented here. It moves from 50 centimeters per second up to 1.2 meters per second. So it gradually decelerates and then accelerates again after passing the constriction. And this is in fact very similar to the results obtained by Srinivasan on the real bee, where you see that within a similar tapered tunnel, the flight velocity, the flight speed here is first reduced and then increases again. How much just finished? I'm finished. So the autopilot automatically makes forward speed decay and grow proportionally to the local corridor width. The main point is that this is done without any measurements of forward speed and distance to the walls. So optic flow measurements are sufficient. So my summary and conclusion. So I've shown you some attempts to link visual and motor systems To close the loop with the environment, Octave and LoRa autopilots consist of optic flow regulator. These are feedback control systems that explain how an animal may navigate in the absence of contact with the ground. Octave controls the ground height, as you heard, and LoRa controls the clearance and the forward speed jointly. Octave autopilots accounts for various behaviors observed in insects, such as takeoff, level flight, terrain following, wind reaction, drowning in mirror-through water, I had no time to discuss about that, and even landing at a constant slope, as observed in honeybees by Srinivasan. The LoRa autopilots may account for other behavior observed by Srinivasan and by ourselves, namely centering and also wall following and speed control. And both autopilots are actually but working hypotheses for us. We constantly plan specific experiments able to falsify these hypotheses, and this gives rise to as many PhD subjects. So these minimalist autopilots produce behaviors without any needs for range or speed measurements. They are simple in terms of neural or electronic processing. They are as appropriate for insects as they might be for aircraft or spacecraft, and particularly small aircraft. And the conclusion of my conclusion is that biorobotics, in fact, creates a salutary synergy between scientific disciplines that unfortunately used to talk little to one another: physiology, ethology, ecology, optics, mechanics, informatics, electronics, control theory, and probably many other, including philosophy. And my second conclusion is that bioinspired robotics is a highly interdisciplinary field, ideally suited for the education of our young students' broad minds. And talking about broad minds, I can just show you our collaborators. Stéphane Violet, who is now running the laboratory, most of these PhD students who are here at this meeting and whom you may have contacted already somehow. Thank you very much.
We started with 10 minutes delay. Nicolas used up all his time and a bit more, so there is space for one or two questions at most.
It's not really a question, but a comment. I was wondering, you showed this video with the male and female overfly, right? And I was wondering how the story ends. Now, the thing is that it seems that they found in 2003 or something that the male overfly moves in such a way that it doesn't induce any optical flow in the eye of the female overfly. So this motion, this following, is just to keep. It's called motion camouflage actually. And what it's trying to do is trying to hide its position.
Yes, this is a completely different system. I don't think it is based on optic flow, in fact.
No, I think I might have something to do, because actually what it's doing is exploiting somehow that the optic, and you cannot measure distance only with your eyes.
Yeah.
So the motion is such that you don't see the male overfly, the female overfly doesn't see the male overfly coming. So, well, it was just a comment because I saw the video and I thought immediately on this.
There was a question over here.
How will I account for the space structure of these patterns?
What was the question?
You can have different space structure of the patterns. For example, geometrical structure of the patterns which move. How will you account for this?
Yeah. So, for example, you may have noticed that we don't care about the actual spatial frequency, because this type of motion detector that we built here operates regardless of the spatial frequency. Although, if the spatial frequency is very high, it will see something like gray, and it will see nothing. But basically, you have to keep in mind that every contrasting border, it can be a gray versus a gray, down to 5% contrast, will give rise to the same output in this elementary motion detector due to the thresholding that is done. It's very different from the Reichert correlator, for example.
We need to stop here, otherwise, to hear again.